The first video in the Ultimate qPCR Experiment series describes how to plan and scope a qPCR experiment and describes the revelations from tissue subsections. The beginning of any experiment, including a qPCR experiment, is the planning phase. Of interest to note down are the biological groups that will be tested in the experiment. So BG1 stands for biological group 1 versus biological group 2, BG2, which could be the control and treatment groups. An experiment may be multidimensional, which could involve not just biological treatment groups, but also time points, T0 versus T1, for example. If I have two treatment groups and two time points, as we see here in parentheses, I will need for each of these, if I'm working with mice, for example, 10 mice per treatment group and per time point. Ultimately, that would require 10 times 2, which is 20 mice to test the two treatment groups, 10 and 10, and 20 mice to test the two time points, again, 10 for T0 and 10 for T1. If I add up the number of biological replicate mice in the experiment, that would be 20 plus 20 equals 40. And then I'm also going to incorporate in my experiment technical replicates. This is the pipetting of typically triplicate replicates of the cDNA derived from each individual biological replicate in a qPCR experiment. Many labs perform 3 technical replicates per biological replicate in the qPCR plate. So 40 biological replicates times 3 technical replicates equals 120 wells required for each individual target being tested, being interrogated in the sample. A target meaning a gene. So let's assume that we have 4 targets to interrogate. That would be 4 targets times 120 wells per target. That would require a total of 480 wells for this relatively small experiment. So a relatively small experiment can require easily 5 to 10 plates, 96 well plates of wells. Working with cells requires less biological replicates per treatment group and per time point. And the total becomes a smaller number, approximately 2 plates in this particular example. Human samples are the most difficult to work with because it's very challenging to achieve reproducible tissue extraction and treatments with humans. Therefore, more biological replicates are required to increase the statistical power in an experiment and minimize variability. So in this particular case, we would need 2,400 wells for the same type of experiment because we need so many biological replicates, hence 24 plates. Scoping an experiment helps in planning the number of reagents, plates, cell culture media, animals, animal feed that would be required for a given experiment to map out the time required to perform the experiment and the associated costs. Time is another issue in an experiment, and often one that is not described in detail. If we measure the transcription of genes versus time in an experiment, it's often found that different genes, in this case gene A, gene B, and gene C, are transcribed at different times over after treatment. So if this is T0 of treatment, gene A could transcribe very early after treatment and then regulate back to baseline. Gene B, as we see here, transcribes a little later in time after the initial treatment and then regulates, and gene C even later. So many labs choose a randomly derived time point to harvest cells or tissue. With cells, it's often 12 hours or 24 hours or 8 hours. With tissue, it could be 24 hours to days. But typically, labs choose one particular time point and often not based on experimental data. So let's assume that this was the time point that was chosen by a given lab to harvest cells or tissue. In this case, if this time point were chosen, then there would be no difference in transcription for any of the targets, A, B, or C. Everything would be at baseline. So the solution to this problem is to perform a time course. And in qPCR, this could be an initial optimization experiment where various time points are chosen post-treatment to harvest cells or tissue to assess where each individual target transcribes. So what we're trying to understand from a time course assay is where the transcription window in time is for each individual target, so that logical time points can be chosen to perform the experiment to assure that adequate data and reproducible data and high quality data is obtained for each of the individual targets in the experiment. In this particular experiment, if we wanted to test transcription of gene A, B, and C, we would need a minimum of 4 time points. You would need T0 as your control, along with a time point for A, a time point for B, and a time point for C, because none of the transcription windows for these 3 targets overlap. Tissue is another interesting subject in qPCR. And when tissue is extracted from an animal, it is often not described in detail how to subsection tissue and the effect of subsectioning tissue on the results. So in this particular example, 2 treatment groups were chosen, control versus treatment. And in the upper panel here, we see that the brain was extracted from the animal and whole brain was homogenized. We'll assume these are mouse brains, homogenized. And the RNA extracted and the cDNA produced from whole brain samples. And unfortunately, the CQ value for the gene of interest was 25 for the control RNA, and again 25 for the treatment sample. So therefore, the delta CQ between these 2 treatments is 0, 25 minus 25, and hence there was no change in expression for the target of interest between control and treatment. However, had the tissue been subsectioned into component subsections, as we see here, what we see now is for the occipital lobe, we actually see a difference in the expression, where we have 23 CQ values in control, and it decreases to 21 in treatment. Therefore, the delta CQ is 23 minus 21 is 2, which would result in approximately a 4-fold increase in gene expression for a target of interest in the occipital lobe. For the temporal lobe, we also see a difference, 24 versus 26. So 24 minus 26 is negative 2, which would result in an approximate 4-fold decrease in gene expression for the temporal lobe. All the other components of the brain gave the same CQ values, which would essentially show no difference in expression for those components. So by subsectioning tissue, we're actually able to glean much more information about our gene of interest pertaining to where the treatment affects expression, in this case, the occipital lobe and the temporal lobe, and the full difference in expression itself. The reason why we saw no difference in the whole tissue extract was simply because by extracting the entire RNA from the whole brain, all the other components of the brain that were unaffected diluted out the effect of treatment in the two subsections of the brain that were affected.